Welcome, everybody. I'm gonna take you on an AI journey. Like most of the things that have happened throughout my career in pathology, I've been doing this for about 40 years now. We don't get really access to these things during our training. So when I started my training, I was an H&E pathologist. Next thing I knew, then comes along immunohistochemistry. Then we've got prognostic and predictive markers that pop up, then molecular testing, first FISH, then PCR, then next generation sequencing. And now we are rotating back into a realm of AI that probably most of us weren't exposed to. Certainly I wasn't during medical school or throughout my residency. So I'm going to take you on a little bit of a journey as to how we got here and why I believe that this is so critically important where we're at right now. So, I love this diagram. I've been using it in my talks for a lot of years, the little caveman pathologist, because really what it tells us is that the true job of a pathologist is to classify disease. And we classify disease based on the therapy options that are available to us. So back before there was biomarkers, when I first started my residency, it was really all about the surgeon. And we delivered things like tumor size, tumor grade, status of lymph nodes, how well they excised the lesion, et cetera. And really our job as a pathologist was we looked at samples grossly and we said, this area looks benign, this area looks like tumor. And then under the microscope, we just confirmed that what we thought was tumor, and we got some new characteristics. And what we thought wasn't tumor. And sometimes we were surprised even by that. Things that looked benign turned out to have cancer in them, et cetera. The one thing that we really learned, though, was everything that looked the same didn't necessarily behave the same. And that's why we have these advanced tests. That's why immunohistochemistry, molecular testing, and AI becomes so important. This is a great slide just reviewing what the power of stratifying disease really meant to us. So if I go back 100 years ago and we looked at things like lymphomas and leukemias, we really just classified those as diseases of blood, and the 5-year survival of those patients was 0, largely because there were no therapy options, right? So we could say you've got a disease and we didn't know what to do with it. Then we got a little bit better and we could decide, well, some of these are tumors of blood, others are within lymph nodes. Then we started being able to subclassify those and relate them to different therapies. And today we've got about 100 different classifications of lymphomas and leukemias with 5-year survivals north of 70% now. So what we do as pathologists and why we classify disease makes a difference in patients' lives. Now, as a pathologist, there's many, many different ways that we can classify disease. So on the left-hand side was more what I learned during my residency, histopathology correlation. So under the microscope, even though this is an example of breast cancer, even though we would issue a report that would say breast cancer, all of those different subtypes of breast cancers confer different prognoses. Unfortunately, those different subtypes weren't highly correlated with specific therapies. So it was helpful to know who to treat and who not to treat, but not very helpful to understand what to treat them with. Now, immunohistochemistry helped us get a lens to look a little bit deeper into the tumor and look at specific protein expressions. So, for example, when anti-hormonal therapy became available in the mid to late 70s, and got into practice in the 80s, it became very important for us to understand which patients expressed hormone receptors. And the way that we used to do that back in the day was with what we call bind and grind assays. At surgery or just after the sample was removed, we would take a piece of tumor, freeze it, then grind it up, and then we would measure the hormone receptors within that ground up piece of tissue. And that was great, except we recognized that that ground up piece of tissue contained benign elements, in situ elements, and invasive elements. And we really only cared about the expression in the invasive elements. So immunohistochemistry allowed us to do that. We could apply specific antibodies to estrogen receptor, progesterone receptor, and then more importantly, in the 90s, early 2000s, HER2 new protein became very important as well. And then we took it a step deeper and we started looking at things like comparative genomic hybridization, and then eventually transcriptomics and next generation sequencing. So in the beginning, we had prognostic markers. That was great. It provided information on patient outcome, regardless of the treatment that they received. And that was largely to tell patients, you're going to do well or you're not going to do well, right? And those, again, were the markers that we already talked about. Now, I like to point out that the sorts of things that we look for today, for example, tumor infiltrating lymphocytes, which are a hot topic today, was first published in 1922 with an observation from Mayo Clinic that tumors that had these tumor infiltrating lymphocytes did much better. Now, of course, there were no therapies back then, no such thing as immunotherapy, no chemotherapy, but it was a nice observation. So we've been observing things in pathology for a really long time that correlate with outcome. And now those observations are coming back and having clinical relevance, especially with the emergence of antibody drug conjugate therapy and immunotherapy. And I'm going to describe that a little bit more detail later on. So for example, in breast cancer, we had this subclassification called medullary cancer that we knew looked very aggressive, high mitotic rates, really pleomorphic cells. But for some reason, stage for stage, they did much better than those that didn't have this dense lymphoplasmacytic infiltrate surrounding it. So again, one more highlight of the immune system attacking a tumor cell, conferring a better prognosis overall. And in colon cancer, we knew that right-sided colon cancer that had intraepithelial lymphocytes, these, I don't know if you can see my mouse, but these little small dark blue cells in the middle of the tumor. And we now know that that's associated with microsatellite instability. And really what we were defining was the patients of colon cancer patients that expressed microsatellite instability, that had a much better prognosis. And we learned later on, didn't really need the chemotherapy treatments of those without those findings. So when therapy options are limited, the job of the pathologist was pretty straightforward: find features and do prognosis. And in the day, even when I started my residency, our options were pretty limited. Surgery being the mainstay, chemotherapy and radiation therapy. But then the world changed. So about 15 years into my practice, the emergence of targeted therapies really came. And what's the importance of targeted therapies? Of course, we all heard about precision medicine and how this was going to transform things. But in essence, what targeted therapy did was two things for us. One, as a pathologist, it now made the oncologist my key customer. So now all of a sudden, the biggest question was, does the patient have a target so I can do targeted therapy? For the patient, what it really meant was the graph on the right. So initially, patients that got targeted therapy got a much better benefit early. But the real problem was, as we extended out, they didn't get the overall survival benefit. So a great progression-free survival benefit, a great benefit from patients in that they actually see the response very quickly, but it wasn't durable. So that introduced the concept of predictive markers, and again, made the oncologist my main customer, because now the question was, what treatment should I give to the patient? Not how well will they do without treatment, but specifically, how should I treat a patient? And that's really the promise of precision medicine. There's a great article from Roy Herbst, 15 years ago now, really telling us that if we really want the precision, the promise of precision medicine, it's going to really involve us doing a detailed analysis of both the tumor and the host, and really understanding the interactions involving the tumor and the host to figure out who's going to be most sensitive to therapy. At that same time, we understood that our emerging hallmarks of cancer, the classic paper written by Hanahan and al. in Cell in the early 2000s, got updated, recognizing that the immune system was an emerging hallmark of cancer, and that to really get cancer, you really had to be able to, the tumor had to be able to avoid immune destruction, that you needed a pro-tumor environment surrounding the tumor as well. So we now have many, many biomarkers, and continuing on with the breast cancer example, it's not just one or two. when a patient gets diagnosed with breast cancer, now pathologists have a lot of responsibility. Now we're looking at not just estrogen receptor, progesterone receptor, and HER2, but now things like PD-L1, tumor infiltrating lymphocytes, homologous recombination deficiency, gene signatures, tumor infiltrating lymphocytes, et cetera. So this is a hallmark of pathology. We have a lot of things that we need to do when we look at a tumor now beyond just what's its size and grade. Now, the game changer in my mind was the emergence of immunotherapy. Now, immunotherapy had been around for a while, but this is really the emergence of checkpoint inhibition therapy, which was allowed to be brought to the clinic and really for the first time showed disappearance of some tumors. So you can look at a patient with non-small cell lung cancer on the far left, a PET scan with all of those black dots representing metastatic disease that 6 weeks after treatment dissolves all of the tumor, no more evidence of tumor anywhere. And here's an example of a patient with a melanoma that was treated with pembrolizumab, and 3 months later, the tumor is basically gone. Now, when we look at the graph of what that actually meant, what we see is we don't get a ton of benefit early on with immunotherapy, but we do get a long duration of response in some patients with probable cure. The issue is it's only a small percentage of patients overall. And so the question for everybody, clinicians, pathologists, et cetera, it's twofold. One, how can we better identify the patients that are going to get that prolonged duration of benefit? And number two, how do we move that curve so that duration of benefit goes from 5% of patients, 8% of patients, up to 80 and 90% of patients? Now, there's some real differences between targeted therapy and immunotherapy, which is, a mind shift change, I think, for everybody. So targeted therapies were really important. They were organ specific. Negative patients didn't get any benefit from the therapy. Their benefits were seen early. The duration, unfortunately, was limited. They had limited impact on overall survival. And our biomarkers were focused on the tumor itself. So everything was about the tumor. In immunotherapy, it's really not just about the tumor. So there's pan-tumor potential. Patients who are negative for the biomarker still might get benefit. And there's some reasons for that. I'm not going to go into that in this lecture, but we could discuss that maybe at a future date. The benefit's not always seen early. You get an extended duration of benefit, at least in some patients, with a strong impact in overall survival. Those are the two big deal changes. And importantly, the biomarkers that we look at now aren't just on tumor cells. They're the tumor cells as well as the tumor microenvironment. And I should add in probably some host factors as well. So this is the graph, this is the cartoon that I made up a couple of years ago that to me really explains and will be like the key reason why AI is going to be so important for us to get to that next wave of biomarkers. So when we had chemotherapy, biomarkers not very important, just prognostic and predictive factors. Targeted therapy, the rise of biomarkers, right? We really needed to understand which cells, how we were going to target the specific cells. And we also wanted to start understanding resistance mechanisms to understand why those targeted therapies would stop working. But once we move into immunotherapy, combination immunotherapy, and antibody drug conjugates, now it's about more than just the tumor cell. It's the tumor cell and the cells that are surrounding the tumor cells. It's about the tumor microenvironment. It's about the patient's host factors that might also influence response to therapies. So my argument is going to be, and I think our issue is always the same, tissue is the issue. We have limited pieces of tissue on patients, right? We're taking smaller and smaller core biopsies. And the question is, how are we going to answer all of the critical questions to understand who's going to respond, especially to antibody drug conjugate therapy and immunotherapy? If it was all about targeted therapies, next generation sequencing has been handling that fairly well, along with some proteomics. But my belief is for us to really move into patient selection for immunotherapy, understand what combinations of immunotherapy to give to specific patients, we're truly going to have to understand the spatial relationships of the tumor both within itself and within its surrounding environment. And that's what spatial biology is really all about, with the goal being to get to that red line at the top. What we really want to do is figure out what combinations of therapies we can give so that we see response rates in 80 to 90% of patients with long-term survival. And to do that, you have to understand more than just the tumor. We have to understand a lot more about the tumor, so don't get me wrong with that. But we also need to understand an awful lot about the tumor microenvironment. We need to understand relationships between tumor and blood vessels and other entities, as well as host factors. So why is this important? So I use as an example, and I'm sure everybody is pretty familiar with Google Maps. In the 1980s, there was a cholera outbreak in London, and there was a massive search to try to identify the source of that outbreak. Mapping technology was just coming along. And just by mapping where the outbreaks occurred, it allowed them to centralize and find the exact point of origin of the cholera. Is basically, I can map out where the things occurred, and I look right in the center, and lo and behold, right in the center is where the outbreak is. So we have the same thing. If you look at the right, here's a tumor with its microenvironment, and you can see that there's many tumor cells, but interspersed with the tumor cells, there's also a variety of inflammatory cells, and in the microenvironment, we also have mesenchymal cells, we have fibroblasts, we have vessels that are growing in. And so understanding the relationship of all of those things becomes absolutely critical if we want to understand why this tumor is there and how to best treat it. So I'm going to give you just a couple of examples of why we think these are really important for checkpoint inhibitor therapy. The graph on the left is from a meta-analysis performed by Liu et al. That really looked at the ability to predict which biomarkers were going to be most predictive of response to checkpoint inhibitor therapy in non-small cell lung cancer. And what we wind up with is we have a bunch of biomarkers that you can see as the three lines sort of to the right. And those three lines have some predictive ability, but not great. So we do things like PD-L1, tumor mutational burden, gene expression profiling. All of them do better than random guessing, but they aren't really predictive of who's going to get the benefit. And in this meta-analysis, which has some issues associated with it, so I don't want to go as a ringing endorsement for the paper, but in essence, what they said was when you combine features, and in this case, they did combinations with multiplexed immunoflourescence, but other people have identified that even combining features like PD-L1 with tumor mutational burden will be more predictive than either one alone. But more importantly, and I'm going to go into this in a little bit more detail later on, is the examples on the right, which were taken from a series of high-expressing PD-L1 patients. And, so for primary therapy, PD-L1 expression greater than 50% in non-small cell lung cancer now receives pembrolizumab as the primary therapy for these patients with pretty good response, but still within that subpopulation of patients, some patients respond and some patients don't. And just looking at the top images on the right, I wouldn't be able to see a difference as a pathologist. They both look like they have some tumor infiltrating lymphocytes associated with tumor. We highlighted those as blue cells. So even for the non-pathologists, you can see they're sort of blue in both images, and they're arranged in different ways. But when you present them as the lower image as proximity scores, how close are those lymphocytes to tumor cells, you can just see in heat maps that those that are very close to tumor cells, which show up as the orange, do very well, while those that show up predominantly as the purplish blue don't do so well. So the patient on the left responds and the patient on the right doesn't. Now, we've also are beginning to see this with antibody drug conjugate therapy. Antibody drug conjugate therapy. So in the Enhertu example, this is data that was presented by AstraZeneca. They've come up with a score that they call a spatial proximity score. That's really the percentage of positive cells that are HER2 positive. But here's the kicker. They also include cells that are positive if they have one HER2 positive neighbor within 50 microns. So I have no way of knowing how to do that as a pathologist with my eyeball. I can see the cells that are positive, but now I need to know the cells that are next to a positive cell and are a tumor themselves. And that becomes very difficult. And you can see the separation of the curves between patients with a high spatial proximity score versus a low spatial proximity score, both in terms of response rate and progression-free survival. Now, my contention is that when people say the words AI and spatial biology, it's a foreign concept to pathologists, right? I didn't get this during training. I'm assuming that there's almost no pathologists that got this during their training. And so, how do we address this? How do we relate it to something that we've already gone through and understand? And I saw incredible similarities between what I went through learning next generation sequencing and what I'm going through learning about AI and spatial biology. So, the sample that we care about in all these cases is going to be the small biopsy that gets taken of the tumor. So it's going to be an unstained section for NGS. On the spatial platform, we have the ability to stain. So that could be an H&E, it could be an immunostain, or it can be a multiplex immunofluorescence stain with up to 100 different markers on it, anywhere in between, 2 markers, 8 markers, 30 markers, 100 markers. Now, the library prep, the purpose of the library prep in next generation sequencing is to identify which areas of the genome we're going to cover. We don't do whole genome sequencing. We decide that we want to look at the EGFR gene. We can decide which exons we want to look at. We can decide whether we want to look at introns, how far we might want to go into those intronic areas. The library prep for the spatial platform is our experiment. It's what do we want to do with the slide that we have? Do we want to do an H&E or an IHC or a multiplex immunofluorescence? So that's really about the coverage. What sort of cells do we want to be able to identify? Because if we want to be able to identify subclasses of macrophages, then I need to do that with multiplex immunofluorescence. If I just hear whether something's a lymphocyte or a macrophage, I might be able to get away with an H&E. Some of the harder classes like natural killer cells, dendritic cells, I might want to mark with an IHC stain. Then we've got the platform for sequencing that's relatively easy, although it does determine the depth of sequencing. And so you need a high quality sequencer associated with a good library prep. And for AI, we need whole slide imaging. So we're going to need a scanner that's going to be able in high resolution or enough resolution to be able to get us an image of sufficient quality that we can do analysis. In the case of NGS, we create a VCF file, which is basically those variants from normal that we identified in the sequence. In spatial biology, we extract thousands of different cellular and spatial features that we've extracted and recorded. The hard part right now that's still evolving in NGS is building up the annotation system so that we understand what variants mean. Are they benign? Do we not know what they mean yet? Are they clearly pathogenic? And we're still building those in the spatial biology realm. So at Nuclei, we work very closely with pharma trying to understand which features are associated with emerging drugs and outcome so that we're basically building up that knowledge base so that when we see features, we know what to do with them. And reporting is very similar. So again, for NGS, right, I'm looking at my little bars. I have to decide which area of the genes am I going to cover? Am I going to cover gene 1, gene 3? As I sequence each one, I don't get complete coverage on each of those. So I'm going to have gaps that I need to have a sequencing depth so that I can overlap and have confidence in the reads that I make. For spatial biology, we also need to understand what am I going to cover. Do I care about tumor, benign elements, stroma, necrosis, blood vessels, etc.? What cells do I want to find? Do I want to find lymphocytes, macrophages, endothelial cells, etc.? In what states? Do I care about the states? If I'm looking at lymphocytes, for example, do I care if they're a T regulatory cell, a memory B cell, etc.? So I also care about the depth, and the depth is really about the confidence in my read. So when I get a read with sequencing, do I believe it, right? Did I see that read in only one read? Is it in 50% of the reads, 80% of the reads? How confident am I? And for pathologists, that's really about the deep learning models that we use. And some of that is, well, what did we call its truth? So if we trained an algorithm to identify a tumor cell, that's as good as and they were trained by a pathologist, it will only be as good, the model will only be as good as the pathologists that were used to do the training. And so that depends on the expertise of the pathologist, the number of pathologists that we use, because everybody will have some biases. Or do we train to multiplex immunofluorescence, where we can use multiple markers to help define cells or cell states? Or do we use transcriptomics to help us define what cell types are? Or do multimodal analysis? However we do it, here's what you get. We're going to get a piece of tissue, and we're going to do two basic sorts of things. We're going to try to identify what areas of tumor are there, what's tumor, what's the tumor microenvironment, what's the adjacent edge of tumor, what's that adjacent tumor microenvironment. And then we want to understand the individual cells that we can identify. What are tumor cells? What are benign cells? What are lymphocytes? What are endothelial cells, et cetera? Once we do all that, you can imagine that we've now created a map. So if you look at the bottom left-hand side, we've taken the cells that were in the upper left. We now know and have identified each of those types. We've just represented them with a different color in the map. And now we can start identifying relationships, densities, proximities, absolute numbers, all sorts of things. And I've got some examples on the right-hand side, but it's basically endless on what we can calculate. So for NGS, we went through this too, for those of you guys that remember. So when we started with NGS, really the only thing that we could identify were single nucleotide variants. Those are easy pipelines. And small indels were relatively easy too. But over the last decade, our pipelines have advanced, and now we have the ability to call larger indels, copy number variants, structural variants. Those are all relatively new, and it took many years for us to hone in on what are the best pipelines to do that. And so when you look at the plethora of pathology features that we can extract from, from these slides, and you can see I'm not going to read through all of them all, but they're very complex features that a human eyeball just couldn't do, right? My eyeball can't do mean percentages and standard deviations and et cetera. But I can break them down into three broad categories of pipeline that I want to know. Tell me about biologic interpretation. Can I predict which patients are going to benefit and which ones won't? Tell me about pathology correlations. Can I predict things like other molecular features like microsatellite instability, homologous recombination deficiency, or simple things like mitotic counts, et cetera? And can I identify cell associations? Now, that all sounds easy, but I just want to highlight two things for you. So way back when I started my residency project was actually the development of the first telepathology system. So I was in the department at Rush. Ron Weinstein was my mentor. He had the idea of telepathology, and I implemented it. So I created the robotic microscope, wrote the interfaces. We got contracts with the DoD, did everything by satellite at the time, and really demonstrated that we can read slides off of a monitor just as easily as we could under a microscope. So we could transfer that. And then we wanted to move into image analysis because we actually start analyzing the image. And so it turns out that's not as easy as what it looks. So for the last 40 plus years, we've looked at different methods of how we even identify a cell and then classify it. And I've just listed here the many different ways that people have gone about it. Just simple things like the size and shape of cells. Other people looked at intensity measurements, how dark is something if you grayscale it, texture patterns. We're using things like convoluted neural networks and deep learning models to try to discriminate that. But there's a variety of methodologies to do that. And even when you try to do it and you use things like multiplexed immunofluorescence, there's some real problems associated with that. And they're just problems of biology, because unfortunately, when we take a section, the cells don't all separate cleanly. They're overlapped. We've got a thickness associated to the slide. So for example, in the top right, we can see a slide that happens to be stained with a cell that's stained with CD3 and CD20. And you can see how that cell is segmented. That's a segmentation error. This is not a lymphocyte that's co-expressing CD3 and CD20. This is an error of segmentation where we took two lymphocytes and called them as one. So one of the lymphocytes is a T cell, one of the lymphocytes is a B cell, and they're just missegmented. But there's no easy way for a pathologist to see that on the slide. We can see that when we apply the MIF. Then you've got things like macrophages that are really, really hard to segment because they've got all sorts of projections. They're not a nice, neat little round cell, right? They have all these little projections moving out all over the place. And so it frequently overlaps in other cells. So you really need intense algorithms, pipelines, just like NGS. The value of those next generation pipelines to do things like copy number variants, large indels, et cetera, needed to be developed. And at Nuclei, we've recognized the same thing, and the pipelines become absolutely critical. So, for example, we've developed what we would consider the next generation pipeline for multiplex immunofluorescence to overcome the problems that you get when you look at an MIF slide. I'm going to go into that in a little bit more detail, but to do that, I'm going to have to take you on a little bit of a journey. So all these things are great. We're doing AI. We've got these models. You hear everybody will start talking about it. You know, you'll read the paper in Nature Communication. Oh, I did this and I was 90% accurate. You know, what does that really mean? How do I evaluate if an AI model is really what I want? And so you can do that really with two common, simple things for everybody to understand. In our world, we call them precision and recall. Precision is positive predictive value, recall is sensitivity. But really what it means is the top two questions. How many of the retrieved elements that you got are relevant? And how many of the relevant elements are retrieved? So on the left-hand side, I've got things that are relevant, right? On the right-hand side of the screen, the false positives, I'm saying those are things that aren't relevant. And then within the circle, those are the things that are retrieved, right? So I'm going to walk you through a couple of examples. So here's an example of a good recall, but bad precision, right? So good recall says, how many of the relevant items are retrieved? So that's pretty good, right? We can see that we've got 2, 4, 6, 8, 10 out of 11 possible things retrieved in the green circle. So that looks really good. You say, how many of the retrieved items are relevant? Not so good, because within the green circle, I also have a bunch of false positives that were included. So this is a great example of really, really good recall, but bad precision overall. I can also have the opposite, right? So now everything that I retrieved is relevant, right? That's perfect. The problem is I only retrieved a small amount of the relevant items that I was supposed to identify. So these are real, it's simple concepts. It's really simple when you present it this way, but just remember those two questions. How many of the retrieved items are relevant and how many relevant items are actually retrieved? And the score that we use to understand those two measurements is what's known as the harmonic mean. And the harmonic mean is a reciprocal of the arithmetic mean. I've done the formula for you there, so you could figure it out. That's exactly why it is. It's the reciprocal of the arithmetic mean. And you do the math, and that's what you come up with. Don't worry about the math. The bottom line is the harmonic mean is the best overall measure of performance. That's the way we measure how the model is working. And we represent that as an F1 score. That's how we call it. So for example, this is from a Cell paper that was performed out of the Stanford group. Data set was available. They had applied clustering based, the standard ways of analyzing multiplex immunofluorescent images, state of the art, published in Cell. So, reviewers looked at it and said, this is a reasonable way to look at the data. They applied clustering methods, and you can look at their F1 scores, which are the blackish bars, on identifying cell types. And other than identifying CD8 cells, they're actually pretty poor. The model doesn't perform very well, right? Overall, if I take all cell classes, it performs at about a 65% level. when we applied our multiplex immunofluorescence deep learning algorithms to that, we perform at over a 90% level. And you can see most of the cell types, our F1 scores are over 0.9. So that's a good thing to ask when somebody is presenting a model to you, to ask what their F1 score is, because that will give you an approximation of how well that is working. So we have things that are very, very analogous to the NGS workflow: preparation, sequence, and analyze. We stain, whole slide image, and analyze. I'd say one of the things that we're missing in the spatial world that we at Nuclei are helping to develop with our academic and pharma partners is the equivalent of the genomic data sets, so that we can understand the knowledge bases to understand what all of these things actually mean. So in the last couple of minutes, I'm just going to give you some examples of why do we care. And this is not an exhaustive list. We'll open it up for questions at the end. This is really meant to just sort of tease you at the ways that spatial biology can really be used. And I'm going to break those down into 4 categories: translational research, patient enrichment for a clinical trial, companion diagnostic development, and what I call pathologist augmentation tools. And these are just going to be examples. So for example, let's say that we've got a phase 2 trial that we're running. And like most phase 2 trials, we were looking for a signal. It's great. We identify a signal in the trial. But like always, there's going to be some patients that got a phenomenal response, super strong signal, right? Tumor behaved unbelievably. There's going to be other patients where the tumor basically ignored the therapy. It just kept on growing. It's like it wasn't touched. And then we're going to have everything in between. One of the ways that people can use the spatial biology is to take samples from the super responders and the rapid progressors, extract the thousands of spatial features and relationships and things associated with those two things, and then reduce the data to the minimum set of features that best distinguish those rapid progressors from the super responders. And now you've got a hypothesis that you can test, right? Now you can go back to the trial, blind those samples again, run your new predictive algorithm and say, could I now find that hierarchy? Could I find those that will definitely progress, those that will definitely show a strong response, but even maybe the gradation of response, depending on how strong the signal that I identified actually drives the influence of the drug. We can also do things like patient enrichment for clinical trials, especially those trials that are largely dependent on a molecular test that take time, effort, energy, and cost to actually perform, with time and cost being the 2 strongest factors. So for example, we've been able to create H&E models that predict all sorts of things, from immunohistochemistry to gene signatures to mutational status. And the idea behind that is at the time of diagnosis, you could flag a pathologist to notify a clinician that a patient might be eligible for a clinical trial. Of course, for the clinical trial, they'll still be registered on the actual diagnostic used in the trial. But being able to predict from the H&E things like microsatellite instability status, whether a tertiary lymphoid structure is present or not, or an interferon gamma signature is present, can all be performed with very good AUCs approaching 90%. So these are ways to quickly employ things within the clinical trial space that can benefit enrollment of patients more easily and quickly. So this is the example that I showed you before. We've actually validated this now with several other cohorts. It was first presented at ASCO in 2021. And again, the idea was, could we take all the patients that received pembrolizumab, all at stage 4 lung cancer, all at PD-L1 greater than 50%, and identify within that one subset patients who get a better response overall to the therapy than others. And in this case, it turned out to be an association of the proximity of tumor infiltrating lymphocytes to tumor cells. And you can see the difference in survival rate between the 2 groups. But more importantly is the little graph in the bottom of the screen, which shows the PD-L1 score between those that got the benefit and those that didn't get the benefit were exactly the same. And actually, the median PD-L1 score, although not statistically significant, was slightly higher than the PD-L1 score of the patients that actually got the benefit. So this is a study that I actually performed back with the first real biomarker that we tried to put out as a companion diagnostic, Enhertu and Herceptin. So when Herceptin got approved, it got approved with a companion diagnostic, Herceptin at the time. And we launched that to the pathology world, trained them up on how to read it, had a scoring guideline. And then we said, well, how well do pathologists score actually correlate with a new surrogate that was arising for predicting patients responding to Herceptin, which was amplification of the Enhertu gene. And so we took 10 pathologists that range from community pathologists up through academic pathologists. So it's really a homogenized group of 10 observers. And you can see in the blue bars where we looked at how well they correlated with fish, the answer was not very good, right? So most of the observers were, actually poor, less than 80%. There were, one that was up around the 90%, that was okay. But the important part of this study was when we gave them an image analysis tool, basically an augmented tool to aid them in the analysis, everybody looked the same and everybody had better than 90% accuracy. So this really raised the question of the possibility of an augmentation tool. And this has now been extended to, could we actually approve an augmentation tool at the same time that we approve a CDX? So not change the world, but say if pathologists read better with an augmentation tool, and we've demonstrated this before, should we be providing augmentation tools as part of pathologist training and as part of the clinical trial so that we can get an FDA label that says augmentation is okay to use to identify patients? And so one is, we looked at a correlation between pathologists scoring a tumor proportion score by themselves versus scoring it with the aid of an algorithm. And you can see there's a correlation, but it's, there's still movement on sides of the line, right? So you can see there's patients with augmentation that score over 50% that aren't over 50% and vice versa. And so on the bottom right, we can see what that actually translates to. And basically what it says, if you look at how pathologists score, that it scores a lot of patients being negative for low expression of PD-L1 that are actually PD-L1 positive. And so that means that there's a significant portion of patients that are being missed by having standard pathologists, and that there's a variety of factors that I won't go into that, as to why that could be the case. But I think the time is right for us to start adding augmentation tools to pathologist scoring that improve the reproducibility, but more importantly, identify patients who could really get benefit from therapies. And the last case example that I'm going to use is virtual multiplexing. So, a lot of these things and a lot of the relationships that we might look at might be dependent on the relationship between different cell types that we need to identify by a series of markers. And there's a very well-defined regulatory path on how we take single-plex immunohistochemical bright field stains through the FDA. There's a little bit less clear path on how we take multiplex immunoflourescence images through the FDA, from getting scanners cleared, kits cleared, training up pathologists, et cetera. So to have the ability to be able to do standard immunohistochemistry, register those slides, overlay them to create a virtual multiplex, and then calculate features based on that are really important. And so it's not just as simple as identifying cells. There's some statistical and registration things that are a little bit tricky to make that happen. But the example on the bottom part of the screen happened to be a 6-plex that we received from a customer. And they wanted to understand associations between PD-L1 and cytotoxic lymphocytes. And we were able to take the 6 individual stains, register them, overlay them, and then through a virtual multiplexing, identify the specific cell types on a projection of a single slide, and then able to calculate features that allowed us to calculate things that would just be impossible for pathologists to do on 2 or 3 separate IHC stained slides. Last is pathologist augmentation. I'm not really going to talk about this at all, other than to say there's lots of people that are looking at these things. The purpose of this would be create an environment that allows me to do my tasks, make it easier to get these jobs done, make it simple, convenient, economical. People will do it. And those are things, you know, the standard things that we always used to do. Mitosis counting, distance from the ink, how close were we to the excision margin, lymphovascular invasion, determining reliably the maximal dimension of tumor, cell counts for placing samples within next generation sequencing, area measurements, pleomorphism, IHC quantification, et cetera. So my last slide, and really what I wanted to project, was this idea away from precision medicine and move into the concept of health intelligence. So precision medicine was really about taking biological data, and that was interesting. We took genetics, we took phenotype, you know, X-rays and histology slides and clinical records, and that was really what we call precision medicine, pull out features from that. But health intelligence is really about taking all of that biological data and applying machine learning and computation to it to really derive insights and outcomes that will finally get us to health intelligence. And so what we're doing at Nuclei is providing that machine learning and computation today predominantly on pathology images, but we also combine that with other data sets that we get to truly give you the potential for health intelligence. So I'm going to end there. Hopefully, Oscar has been accumulating some questions. I think we've got a few minutes left, and I'll be happy to take any questions that you've got.
Thank you, Ken. Thanks a lot for an excellent presentation. And you have the Q&A chat where you can post the questions. There's one question from the audience in that the field will advance as long as we share enough data to build better models. Models will increase accuracy and depending on how data is shared. Do you think the current framework of patient privacy laws allows this? So in your comparison between NGS and spatial biology, do we need to make any changes to the current privacy laws to allow sharing data, or the current framework is enough?
So, you know, patients always have the option to share their data. So the current privacy laws just require that a patient consent to you know, have their data shared. I think as people understand the power of these tools, there are going to be enough patients in the world that will consent to share that information because it benefits people. I think people with cancer especially want to help other people with cancer. They know it affects their other family members, that their family is at risk. Of course, it would be fantastic to change some of the medical privacy laws to advance this for the health of the world, really, right? I mean, if we could advance these things faster and learn faster, I think that's of utmost importance. You know, it's the first part of the question, I'm not sure is 100% understood. And that's how much data do we really need to acquire before these models can really sort of self-learn? And, you know, a lot of things that we do in pathology is you see papers published with a couple of hundred cases. We've seen some of the approval processes. The first cleared process had 60,000 patients within their cohort. You know, while I was at Human Longevity, we were trying to do similar things with next generation sequencing data and say, could we learn this AI self-learning model about these things? And I believe that our projection at the time would be that it would have required millions of samples. So it wouldn't shock me before we really get to this point of true self-learning is outcome on millions of patients, right? So that might be 20 years away. So that's great. For right now, and as I try to stress in the talk, we really need to build knowledge bases for spatial biology, and the consumers of whatever company you're going to deal with and algorithms you're going to deal with you need to understand their performance, right? So garbage in, garbage out still holds. If what you call tumor cells or what you call tumor area isn't, what you call necrosis isn't, what you call stroma isn't, then all the calculations are just noise. And so it's really how good are the models? And then we'll do what we can do. We're always going to get, I guarantee 20 years from now, we'll be way better at this than we are today. But you got to start today.
Right. Another question is regarding the spatial proximity score that you presented and the difficulty by the pathologists to implement that in the clinic. What do you see are the current barriers for implementation of something that's just like purely AI-driven? And where do you see the field moving in this space in terms of implementing this in the clinic?
So I had the pleasure of being one of the co-founders of Clariant Labs, which was a cancer testing labs, one of the largest in the United States. I was the founding medical director and chief medical officer. And so as part of that, I had to validate everything that went through my lab. And for those of you that know Clariant, one of the things that we did was we gave tools for ER, PR, and HER2 assessment. And to do that requires an end-to-end solution. So one of the problems with AI is what pathologists do as a test isn't AI. It starts from the sample, how we process it, how it's cut, how it's stained, how it's scanned, and then the algorithm that we apply to it. So I think that this is something that regulatory agencies are going to struggle with. As a pathologist, if I had to validate it, I'm not validating an algorithm, I'm validating the entire test end to end. So that's one part that becomes really, really important. Now, the spatial proximity score is a score, to be fair, it's not my score, totally derived by AstraZeneca. I've seen them present it now a few times. And the significance of that score is that you score not only positive tumor cells, but negative tumor cells that are within 50 microns of a positive tumor cell. I don't know how to do that with my eyeball, right? I don't know how to, yeah, I could get 50 microns as a 20x field or 40x field. There's no way I can go over every square millimeter of the slide in a stepwise fashion to do that, but that's exactly what AI can do. It can classify every cell, understand the relationships between every one of those cells. So I could do it painstakingly. I guarantee you no pathologist would do it. Probably take me 10 hours and my eyeballs would fall out. Or I can use an augmentation tool that just does it. Now, I'd have to believe the augmentation tool. So again, it would have to be validated to the stain, the scanner, the algorithm, the whole thing. And I think that's the problem, is that you always have to think of these things as an entire test.
Yeah, makes sense. Thank you, Ken. A third question we got is, you hinted the version 1 of the H&E models are based on human annotation, and kind of hinted there's ways to improve this. And specifically the question is, can you elaborate on how multiplexing immunofluorescence can be used?
Yeah, so I've done a fair amount of annotation. So I'll just give you the little anecdote for the pathologist there. So one thing that people give me frequently is they'll give me a region of interest on a slide. And they say, okay, here's a region of interest that basically fills up my computer screen. And they say, annotate every cell in the field. And the bottom line was, when I get that, I'm like super confident on probably about 30 to 50% of the cells. And then the other 50% of the cells, I'm not so sure about. It's like, maybe it's a macrophage, maybe it's a fibroblast, because we're not looking at whole cells. We're looking at 5-micron slices that get tips of cells and overlaps of cells. Sometimes I'm not even sure it's one cell. It might be 2 cells that I'm thinking are one cell. So that's an inherent problem, I think, with pathologist classification. And a different approach would be to give an ROI and just help pathologists annotate those cells that you're 100% sure what they are. Right. And then we would get very good at annotations of things, my belief is, things like tumor cells and lymphocytes, because those are pretty characteristic. We can identify those. We'll do less well with things like fibroblasts and, myeloid series cells, dendritic cells, macrophages, because they've got projections in weird shapes. So when you apply things like spatial transcriptomics or multiplex immunofluorescence, and I now say at this XY coordinate, I can see that transcriptomically you are a cytotoxic T cell, or I can stain with a variety of MIF stains, right? And I can layer those MIFs as much as I want. So for example, if I call something a CD8 cell, is it just is it only a CD8 positive? Or do I really call it, do I want to see that it's CD3 positive, CD8 positive, CD4 negative? Now I've got more confidence in my call. And all of these things, I think, are about the confidence in the cell that you detected, in the state that you called it, because more confident decisions lead to better outcomes, because any garbage that gets in just becomes problematic. So I'm a believer that over time, as these new systems develop and we get transcriptomic data, high-plex data, whether it's MIF, MIBI, any of the new emerging technologies, it will make us better as pathologists. And I'm going to tell one story. I know we don't have a lot of time left. I'm going to tell one story of immunohistochemistry. So before, I was there at the beginning. So before we had immunohistochemistry, we used to diagnose undifferentiated malignant neoplasms all the time. And when you did that, you would send it off to the experts in the world. The best expert in the world in the day was Juan Rosai at Memorial Sloan Kettering. Famous studies with him where if we sent off all of our things, we went back, and once we got immunohistochemistry, just basic immunohistochemistry, cytokinin, vimentin, LCA, S100, are you melanoma, lymphoma, sarcoma, carcinoma? What we found was that 50% of what the expert had classified by H&E was wrong. Now, that didn't mean it couldn't be classified by H&E. Once we got the immunostains and understood how to classify, then we adjusted criteria. And within 5 years, the H&E was back up to over 90% on the H&E, because we now knew what to look for. So to me, it's absolutely critical that we start extending our models to higher plex. Just to put it another way, get more confidence in what your model is doing. That higher that F1 score gets, the better you're going to perform.
And the last question is actually on those lines. How to use commercially available spatial platforms like Cosmic, Senius, PhenoCycler. They have multiple layers, orthogonal layers of data. How are they being used today?
So a little bit outside of the scope of nuclei and our talk, right? Because what we do is we're really focused on tissue-based imaging. What we are starting to see more frequently is patients want to combine the spatial features that we extract from H&E, IHC, low-plex MIF with the data that they've derived from these other systems, which is very, very rich. The beauty is, as long as those systems provide an XY coordinate of where they were, we can take that data and overlay it to the XY coordinate of what we do. And one of the things that I should mention, because I didn't really go over it in this, but everything that's done at nuclei, when we extract these areas, when we extract individual cells, everything is exported as a polygon shape with an XY coordinate, so that not only us, but you, if you've got a good bioinformatics department, you could take the output of what we produce and overlay it on top of any of these other systems. So I'm not going to comment on which ones I like the best or what might be the best, because it's problem dependent, frankly, right? You have to understand your problem and then pick the best solution.
Yeah. Thank you, Ken. We're out of time. This is a great presentation. Thank you very much.
Thanks, everybody.